Hi, I'm James Reeder, and welcome to the American Institute of Biological Sciences BioScience Talks, which is a forum for integrating the life sciences. For today's episode, I'm joined by DeeAnn Reeder, who's a professor of biology at Bucknell University and a researcher at the Smithsonian Institution. She joined me to discuss a number of topics, including bats, disease ecology, and community outreach. And underlying all of that is a very important message about the One Health concept, which we'll hear more about in a few moments. And I would also be remiss in failing to mention that One Health is a topic that's of great interest to us generally at BioScience, and so much so that we're actually in the early stages of planning a special issue on the topic. If you work in that area, there's a link in the show notes with more information for potential contributors. But for now, let's go to the interview. All right, thank you very much for joining me today.
It's my pleasure.
Okay, so today we're going to be talking about, One Health, bats, and many other topics. I was hoping you could start us off by giving us a little bit of an introduction to the, kind of One Health concept. How does that approach work?
Sure. I think One Health is a big umbrella approach, and it can be sometimes more narrowly in scope and sometimes really broad in scope. For me, what One Health means, or a One Health perspective means, is that it's an understanding that human health and ecosystem health and wildlife health and even domesticated animal health are all interrelated, right? So we can't just work on one of these aspects in a silo. For example, a human public health in a developing nation or in some place where a particular outbreak is happening, right? We can't work on that in silo without also including an understanding of what's happening in the ecosystems around them and what's happening with the wildlife species around them. Everything is tied to everything else.
And, is the challenge there the fact that, we have systems set up to, graduate scholars, et cetera, fill the ranks of the civil service with people who, are specialized in different tasks?
Right, absolutely. So we go down these narrow paths, each individually, particularly as academics, but it's really, really important to maintain some breadth. And I think that the way that we do that these days is to collaborate widely, right? So when there is a project in some place, for example, some of my work is in Uganda and some of it's in South Sudan, right? We try to include the perspectives of, and sometimes the actual academics of anthropology and veterinary medicine and public health, right? So we have to create these interdisciplinary teams so that the expertise of each of these different components of the One Health concept are well represented. So I think that's how we deal with that. We are siloed and everything's becoming very reductionistic, but by collaborating broadly and maintaining an open mind, I think we can keep that One Health perspective front and center.
Okay, and how does that come into play? I think probably most of our listeners are more familiar with the ecology side. How does it come into play that ecologists are relevant to things like public health, for instance? Sure.
So ecosystem health is something that we consider in the modern era in terms of what it is that humans, for the most part, are doing to the landscape, right? So we are degrading habitat. We are sometimes completely destroying habitat. And the animals that lived in that habitat have to either move or go extinct or do something different. And people are now moving into those places. If, for example, I'm studying the ecology of a particular bat, and I might be studying its life history, where does it live in the rainy season or the dry season? Which of those is it more likely to be shedding a virus, for example? Those ecological variables have direct human consequences, because if we're trying to avoid spillover of a pathogen into people, right, then we need to understand what's happening ecologically with the animals. We need to know, when is that risk the greatest? So in the rainy season, can I deploy some sort of measure that helps protect people from disease at that particular time? When we think about ecosystem health, right, we're really looking at, how intact are the forests? Are they able to sustain the individual species that used to be there? Or the changes that we have caused now, meaning that animals that didn't used to interact are now interacting, right? So animals, maybe their habitat has been destroyed and they have to figure out where it is that they live now. Classic example comes out of the Nipah virus story in Malaysia, where you have destruction of habitat where flying foxes, these giant pteropus fruit bats, lived, and you have agricultural move in. And so you've dramatically altered the environment in the ecosystem. You have moved in an animal, say pigs, for example, that didn't used to live there. And the bats that used to live there, some of them decide that they're still going to, despite the fact that their whole ecosystem is now destroyed. They're going to hang out in the mango trees or whatever it is that they can find something to eat. And now all of a sudden, because of these ecological perturbations, you have bats and pigs together. And bats and pigs didn't used to be together, at least in that particular place. And now you have a virus that the bats have had for thousands of years and have co-evolved to live with that. And it in that particular place, that virus would not transmit directly to humans. But now there's a new bridging host that is able to get the virus from a bat. And then it itself, so the pig actually gets sick from this virus and passes it to people. So all of a sudden, because of this activity that you had, right, of cutting down forests and bringing in agriculture and bringing in domesticated animals, you've completely changed the dynamics that were in place for thousands of years in terms of the balance that existed between animals in that environment. And there all of a sudden now is a very direct human health, public health relevance, right, to ecological change.
And that's fascinating because if you didn't have, expertise in land use change, if you didn't have expertise in veterinary medicine, agricultural expertise, public health expertise, human health expertise, and ecological expertise all together, in the same room, virtually or otherwise, you wouldn't necessarily be able to address the problem holistically.
Right. And you need to put all the different pieces together. And sometimes you need the different experts in the room to be able to put the different pieces together. One of the first clues when Nipah virus initially came on the scene, one of the earliest clues, right? So it was pig farmers who were getting sick. But within the community, the people who were not getting sick were Muslim because they don't eat pigs.
Right.
And so, it takes maybe a sociologist or a public health official to say, who are the people who are getting sick, right? And how does that factor into helping us understand pretty quick, because they needed to understand really quickly what was happening, why it was happening, and how they could put a stop to it.
Yeah, is it beneficial to have these, sorts of collaborations in place, worldwide, in advance of having an issue?
Right.
Obviously, it's better not to have to figure everything out all at once. But how do these collaborations work in practice? Well,
I think it really varies geographically, right? And even in the best developed situations in which we think we have everything in place, right, it doesn't always work the way that it should, right? So when we think about disease outbreaks, for example, what we really want to be doing is stopping them at the spillover point instead of reacting later once they have already occurred, right? But you have to have various agencies and expertise in place. And this expertise exists all around the world. It exists in developing nations. They have veterinarians and they have public health officials who know their systems well, who know the different agencies within the government. And so the trick is really making sure that there's enough crosstalk between them and that there's communication between them, right? So that if there is something happening in one part of the country where there is some novel sickness that is appearing, right, that information gets to the right people in a really quick manner. The UN has developed some initiatives over time that have made significant progress in that, but I think there's always more work to be done.
Absolutely. Let's talk a little bit about Uganda and South Sudan. What have your efforts in those areas been like, and how has that kind of played into all of this?
Sure. So again, under this sort of big broad brush umbrella of One Health, some of my research interests are just in mammal biodiversity, and particularly in bat biodiversity. I want to know who all of these animals are and what they do for a living. I'm just fascinated by bats. I think they're sort of evolutionary superheroes. And so some of my work has just been conservation based and trying to describe the biodiversity that exists. In my work in South Sudan, right, that's a place where people hadn't worked for decades because of continual conflict. And so there are occasionally these windows of time in which you can go in and work. And I established, sort of friendships and collaborations with South Sudanese and with the wildlife service there. And we are, collectively trying to document what they have. And some of that work is not just bats, but I had some chimpanzee surveillance work that we did and some forest elephant work and really trying to understand the resources that they have in terms of their wildlife. But it also happens in a particular context. So I'm working, for example, in some game reserves on the border with the Democratic Republic of Congo. And the people in that region are part of the Azande people. And so I'm trying to understand, well, what do they know about wildlife? What do they have to teach me about wildlife? They are pretty significant consumers of bushmeat, of wild meat. And so there's some risks for them in that context. So I ask them questions about their disease burden. We talk about some of the risks that might be associated with eating wild meat and how they could potentially mitigate those risks. And we're trying to sort of work in a very holistic way with them. So partnering with Fauna and Flora International in that community, we have rolled out some human livelihood programs. So we're trying to decrease pressure on wildlife and decrease human-wildlife interaction where that disease spillover happens, right, by improving the quality of their life. But it's a very, very rural environment, and many people are, they're living in the forest. And so that's the most sort of intense type of environment in which you really have people living very close to animals. And it's all a part of, I can't just walk in there, do my biodiversity survey where collect some bats, bring them back to Smithsonian and not care about the people there and about the ecosystems there, which are relatively intact and have amazing conservation capacity. And I care about the health of the people in that region. And it's just an amazing place. It's been really challenging, the work. I had to shift much of my work to Uganda because of continual conflict in South Sudan, which just some of the work just became untenable to be able to do there. But I still remain as involved as I can be. And it's sort of a perfect example. And when we ask people about their disease burden, it is extraordinary.
Yeah.
And I ask questions about, have you had exceptionally high fevers with a rash and vomiting and was there, any sort of hemorrhagic symptoms? And the answers to those questions are yes. And so, I would love to get in and be able to study some more of that there. But it's sort of touch and go in terms of safety of the team. In Uganda, a more developed country, we're working in the northwest corner, close to the Democratic Republic of the Congo and close to South Sudan, and still working in some very rural environments. In that context, we're specifically studying the bats that are what we call synanthropic or peridomestic. And there's those ones, like I talked about earlier with the Nipah virus story, that when you encroach on their habitat, they stay. Right? So many times if you destroy habitat, some of those species just go locally extinct. They cannot live in an altered environment. But some species are a lot more adaptable and more resilient, and they are happy. I don't know, it's not the right word, right? But they, it's fine. I'll stay in your papaya tree and your mango tree, and I'm going to hang out and eat fruit and drop that fruit onto the ground where your children are playing, right? So these species that are living really closely with humans and interacting with them, I'm really interested in those species, and I'm interested in the sort of human perceptions of living with those species. I have a colleague, Dr. Imran Ajotre, who's a Ugandan scientist, and during COVID, when we all got locked down, and he was locked down in Uganda as well, and within his district, and he said, what can I do at this time to sort of to work with folks who are very scared, and there was a lot of really negative messaging about bats going on at that time. And because of the links with COVID-19, right? Because of the story that the evolutionary origins of COVID-19 are likely in a bat. And so that gets hyped up by the media. Bats become sort of epidemic or epidemiological villains, if you will. And across the world, bats were being killed because of fears that were stoked by the pandemic. And so Dr. Ajotre, he went up and he heard about some efforts that were about to be undertaken to do a massive cull in 2 different locations and kill, hundreds, if not thousands of bats. And, so he grabbed his little One Health toolkit and off he went to meet with officials in these different parts and to do some sensitization and talk about bats and the roles that they play and what pathogens we think the bats in that region may or may not carry, and when we think they might spill those over or not, and how to protect people. This lovely book called Living Safely with Bats. It was a product originally out of the USAID Predict projects. And so he did this sensitization, but he also asked people what they thought about bats and what their perceptions were. And I think it's only when you work within that context and you understand the cultural beliefs about bats and what people know about disease or not, that you can, I think, start to affect, some change. He sort of looked at it by different tribes, different, sort of cultural perspectives in the region. And some of them were much more bat friendly than others. And many of them were not bat friendly at all. And we see that, across the world. But that was a great, he was able to take his expertise as a Ugandan from that place, right? These are his people. And he has a, inherent respect from them, in that way. And he, enacted significant change. And he was able to, for example, there was probably a few thousand epileptic fruit bats on the grounds of a large hospital in the area. And we've trapped there. And I've seen those bats and seen that place. And the bats were in the place where everyone was waiting to get into the hospital. The bats were in the trees above them, and they were, defecating and urinating on them and dropping fruit. And if you think about, a pathogen, everything has pathogens, right? And so if you think about pathogens, they tend to be, they can be excreted in feces. We know that for coronaviruses. That's a major excretion route from animals. They can be being shed into saliva. And so, if a bat eats a mango, that mango falls to the ground, a child picks up that mango and eats that mango in just the right time frame, and you could have a disease transmission event. And so, they were able to sort of pull together, the government provided some funding, and they built, a covered structure for people to wait under. Problem solved, right? But it took him coming together and the public health officials from that district, so he's, meeting with different government officials, meeting with the hospital officials, and they were able to enact a solution that was local. It was, sort of culturally acceptable, and it saved thousands of bats from being killed. And these bats, these fruit bats in particular, are really critical in terms of maintaining ecosystems. And, they eat fruit and they distribute the seeds from those fruit quite some distance away as it passes through the gut. So they play really big roles. So I was really proud of the work and excited about the work that he was able to accomplish. And likewise, he saved the colony of Eidolon helvum, the straw-colored fruit bat, up in a more northern region. There's thousands of them there, and he saved that one from being completely decimated by working with the locals.
Yeah, that's fascinating. And what generally, was the finding like around attitudes toward bats? You mentioned that they're varied among different groups, but is that driven by past experiences with catching hemorrhagic fevers and such, or what kind of goes on?
I think well before our understanding of zoonotic diseases, right, and of the role that bats may or may not play, right. So all animals carry their own pathogens, right, and rodents carry hantaviruses, but so bats are just one type of wildlife that has some pathogens that can transmit to people, but for centuries, millennia before then, right before we knew anything about the infectious disease, people had attitudes about bats. And I think that in general, people are afraid of and don't understand animals that come out at night. So we see some of the same perceptions, for owls, for example, right? So they're harbingers of evil or a bad omen. A notable exception is in China, where they're considered to be good luck. And so that's a really fun story, and you'll see a lot of imagery of bats in Chinese art. But within Uganda, he was able to find there were folks that knew a little bit more about bats and that understood the sort of messaging about ecosystem services and the role that these bats might be playing in the environment. But many of them said there's two kinds. There's probably close to 100 species of bats in Uganda, but most people will say, well, there's fruit bats, the bigger ones that they see in the trees with big eyes, and they know that they know where they are. And then there's a little insect eating bat, right? And so they're amazed to see the diversity of them, but they generally considered them to be, sort of dirty, unclean animals, and some fear. And there's a lovely story that was relayed to him about sort of the concept of bats as tricksters. And we see this come up sometimes in other cultures as well. This notion, in fact, there's even like 2 Aesop's fables that deal with this. So there's this lovely story about how the king of Buganda, the original kingdom in the southern part of Uganda, he wanted taxes to be paid by all of his subjects, and that included animal subjects. And so, but when it was time for the bats all the mammals were having to pay taxes to the king. And when it was time for the bats to pay taxes, the bat said, "No, I'm a bird." And then when it was time, then they started, collecting taxes from the birds. And when so then they came to the bat, and the bat said, "No, I'm a mammal." And so you see this like bat as trickster. And so to this day, supposedly, right, the Bugandans don't like bats because they fell out of favor with the king for trying to trick him. But we see similar stories, and I think it gets around this notion of, are bats birds or mammals, right? I have been in the field in both South Sudan and Uganda. We might be trapping in a small village, and so we attract a lot of local people because here we are, these crazy, this crazy team of white people and black people, and we've got these nets up, and we're collecting animals not for eating. And I always, if I can, take a minute to have a fruit bat in my hand, for example, and I'll take it over and grab one of my Ugandan students and say, "Come help translate for me." And I'll talk about them. And I remember once I had a female fruit bat, and she had a little pup attached to her. And this woman looked at me and recognized it's nursing. And she said, "They're like us." She thought they were birds, right? But then she saw that connection point. And so we try to do our best to kind of make those connections all the time. But it's these long entrenched cultural perceptions. And I think we have to work really early on all across the world to try to combat those notions. I mean, it starts in kindergarten, right? So I sometimes go to, a third grade class. In fact, I go every October to my local school, and we talk about how amazing bats are. And I think we have to get people to understand how extraordinary they are, and wildlife in general, right? We tend to be afraid of things that we don't understand.
Right. That makes sense. And, what's the kind of practical benefit of having improved attitudes toward ecosystem players like bats? What does that allow us to do that we wouldn't be otherwise necessarily able to do?
So I think there's a couple of things. One of those is just purely about conservation, right? And so I would say that the biggest conservation threat to bats in general is habitat destruction, and so that's a much harder thing to deal with, right? Because there's so many layers to that, right? And people have needs, and everyone needs to eat, and that's a hard nut to crack. But I think one of the other major conservation concerns for bats is disturbance. So, for example, in caves, people will go disturb bats because they just don't like them, right? Or they want to go in and harvest guano, but and I have seen people just throw rocks and trees at bats just because they don't like them. And so I think that if we can get people to appreciate them, we can alleviate some of the pressure on bats. We also know that when animals are stressed, right, their stress hormones go up, and some of that stress may be from disturbing them and repeatedly. So if a bat is having rocks thrown at it and they're having to repeatedly fly from roost to roost, and that's happening every night, right? Or someone is coming into their cave and chasing them away on a regular basis, that affects the bat's sort of, physiology over time. And we see these stress hormones can increase. When stress hormones increase, immune systems become dysregulated in some ways. And we sometimes see immune suppression, which then could lead to increased viral spillover, right? So healthy bats are really good for healthy people. And I would say that about, all sorts of wildlife. But so some of it is a pure conservation message. And then the other part is, to learn to live safely with bats and to live safely with other wildlife is to try to keep the health of those animals really paramount.
And I'm wondering now, do bats get a bad rap as disease vectors? Obviously, we've spoken about them quite ad nauseum, but in relation to the COVID. But are they more likely to transmit diseases to humans or other species, or less?
So that is the question of the decade. Okay, right, and I don't think that it is fully settled, but I think we're getting some consensus on it. So again, I always say, like all other species of wildlife and all species of animals, including humans, right? Bats carry diseases. Bats carry viruses, bacteria, they have parasites. They have They have all of the things that all other animals have, right? So the first question is, do bats have any more viruses than any other group of animals? And in part, that is hard to assess because the survey work has been biased, right? So if you think that bats are full of viruses, you go do virus hunting in bats and you find, if you look, you will find them, right? But we know, so, take a look at water birds. Those are the reservoir hosts for the influenzas, right? Take a look at rodents. They are the reservoir hosts for things like hantavirus. So we can get these really challenging, nasty pathogens that can spill over to people. We can find those from all sorts of animal groups, including livestock, right? But then the next question is, okay, so maybe they do or don't have more viruses than any other lineage of animals. But it does appear that they actually tend to share them with each other more frequently than other animal groups might. And that is because they literally hang out together with other species, right? So if you have an area that has, say, 10 species of rodents, all in that same area, so they're what we call sympatric, they aren't huddling together in a little nest. They each have their own little place in that forest where they are living. If you have 10 species of bats in there, 5 of those species might be together in a cave, co-roosting, and 3 of those species might be hanging together in a fruit tree. So when they are in such close proximity, they are able to pass pathogens, including viruses, between them more so than other animals do. So we know that bats tend to be sharing viruses. There's some good evidence for that. They share viruses across species more than others do. The big question is, are they any more likely to carry these high consequence viruses than other animals? And that's where it becomes really challenging. And that's where I think the answer in part is yes, but only in a few instances, right? So we can't, there's 1,474 species of bats, right? We have a few examples of bats carrying very lethal pathogens or pathogens that are lethal once they spill over to people. We need to understand those, and we need to understand how bats survive those pathogens and when they're most likely to spill over to people. It's really controversial. There's this idea that, because of bats' evolutionary history, they're set up physiologically a little bit different from other mammals, and immunologically, they're different from other mammals. And I think there's increasing evidence that those things are true. So, bats are the only mammals that truly fly. That makes them ecologically unique and it makes them physiologically unique, right? So they have to fly all night long, or for 6 hours a night, while you're pregnant, right? Or while you're lactating. The energetic consequences of that are massive. So it means something different to be a mammal that flies than to be a bird that flies, for example. And so their energy needs are huge. There's this notion, very controversial, it's called the flightless fever hypothesis. There's this notion that, when bats fly at night, their metabolic rate is so high. We know this. There's this notion that their body temperatures rise, that they literally have a fever while they're flying. There's some evidence to suggest that might not be true. There's some evidence to suggest that it might be true. And maybe it's not true in all species. But it's really hard. If you're generating, imagine running a marathon, right? Your body overheats. Yeah. And, and they've got these wings, so they can be losing some of the heat across the surface of those wings, but they're still likely to be overheating. And that overheating and that the metabolic sort of costs of running a marathon every night, which is pretty much what they're doing, right? The costs of that can be really detrimental to our bodies. So they can cause oxidative stress to our cells. That can actually lead to premature aging. And so there's these ideas that people are trying to put together, again, experts in different fields, right, are coming together and sort of connecting all these dots. And there's this idea that the bats are, because they're flying every night, because they've got this metabolic damage that's happening, bats have evolved mechanisms to mitigate that damage that's happening. So they can repair the DNA. So for example, when we have oxidative stress, our DNA gets damaged. And that can lead to cancer, and that can lead to premature aging. Bats have evolved mechanisms to mediate the effects of that stress. We think that's one of the reasons bats live a very long time. 30 years, 38 years is, I think, the highest number now. And so these are not flying mice that give birth to a ton of babies and live 2 years. These are extraordinarily long-lived animals, especially for very small animals. So they live a long time. It's probably tied to being able to mitigate the effects of that metabolic stress. They also have been very rarely ever documented to have cancer. So here's this interesting thing. And then we think that that's also tied to the immunological challenges, that the ways that they have evolved over time, some slightly different, sort of tweaking little components of the immune system so that they're interacting with pathogens in potentially different ways. Now you take a pathogen like the filoviruses, like Marburg virus or Ebola virus. And we know very well in the Marburg virus system, we know what bat is the reservoir host for that. Finding the reservoir host for Ebola is the holy grail, and we're not there yet, but a lot of people are working on it. But you take one of those viruses, for example, and normally when we get a virus, we spike a fever. It's what we do. That helps our immune systems fight that. It has a lot of good reasons to have that fever, and they help fight that pathogen. But if you are, imagine you're a virus that is co-evolving in a bat, and it sort of moves through the population, and you get new pups born, so you have newly susceptible animals. So it persists in this population. Those animals are having a high fever, at least a relatively high temperature every night. And so you've evolved to be able to persist in that environment as a virus. So you're used to a fever. All of a sudden you spill over to a human. Human cranks a fever. Virus says, I know how to handle this. I evolved in an animal that has a fever on a regular basis.
Gotcha. So you've had a problem.
No problem. Right. And so the thing that, so a lot of my work in Uganda now very specifically is about trying to understand the bat immune response to Ebola. And so I'm partnering with Dr. Ken Field, who's an immunologist, and we're doing these immune challenges in these flight tents that we've set up. It's really amazing and interesting work. But we're trying to figure out what it is how these animals are responding. And one of the things that I think is happening with bats, when they have these really high consequence pathogens, they're really good at mitigating the effects of what that virus sort of might otherwise do to them. So when you think about immune responses, I like to think about it as like having your foot on the brake, the gas pedal and the brake at the same time, right? And so you're infected by some pathogen. These are all animals, including us, right? You're infected by some pathogen. You need to you need to activate immune processes to start mitigating the effects of that infection and try to clear that infection. And then you have these other things. So you have something like, we hear about all the time about inflammation. That's something that response can help us fight off pathogens. But sometimes our immune systems go a little haywire. And they have what I call an overly exuberant response, right? So sometimes we call them cytokine storms. And that's what's happening when people die of COVID, when people die of Ebola, they're dying because their immune system's completely freaked out. And they went way overboard and they caused this runaway inflammation. And so it's the people who survive, you get to about day 5 or 6 with these viruses and you go one way or the other. Right? Everything's kind of in check and you don't have too much inflammation and then you start to recover, or you've had this tipping point and you go overboard and you die of a overly exuberant immune response. And bats know how to put those brakes on really, really well. They know how to keep that inflammation in check. And so people have shown that with a variety of different viruses. And we're now also working with our fruit bats on a coronavirus that they carry and how they're keeping inflammation in check when they have that. And so I think we have so much to learn from bats about how they manage viral infections in particular, maybe also bacterial infections and parasites and things like that. We have a lot to learn from them. That's going to inform spillover risk to humans, but it's also just going to inform us about what the immune response is capable of. And it might even allow us to develop some targets, right? So if we can understand that bats are keeping inflammation in check by modifying the expression of gene X or gene Y, right? And there's a particular protein that's being made or not made, then you could imagine a pharmaceutical intervention, right? And so for me, it's about the bats, but it's also about keeping people healthy. And it's about potentially, bats might actually have something amazing to teach us about how to survive infection.
Yeah, the evolutionary superhero moniker is making a lot of sense now.
Yeah.
I'm wondering, about how this is actually studied. Perhaps most interestingly in the field, how what is that experience like of being out there? You mentioned flight nets, I believe.
Sure.
How's that go? Well, what's that like?
Oh, so it has to start with getting the bats, and so we set up. There's a couple of different ways that we can trap bats. There's something called a harp trap that literally looks like two harps next to each other, and it has the little strings, and the bats can't sort of quite get through them, and they fall into a little bag. There's also something called mist nets, which is what people are probably more familiar with. It's what birders use as well. It's like a giant hairnet strung across, a forest. And so that's primarily what we use. We use these mist nets, and we go out and we capture the species that we're looking for. And we catch some other things, and we show everybody, and we say, this is amazing, and then we let those ones go. The fruit bats we bring into sort of semi-captive facility that we had built at Mooney University, and with great thanks to that university. They've given us lab space. We built a molecular biology lab there. We've gifted all the equipment. It's all paid for by the National Institutes of Health. And so we have built this facility. We have a lab, and we have this, locked in, gated 2 bat buildings that are open to the air, but the bats can't get out. And then we keep these tents within them, and we bring the bats in. And for the fruit bats, it's relatively easy because we just go buy fruit in the market, and they figure out real quick how to eat. And so we keep them. They've got they've got a little microchip like we do for our dogs and cats, so we know who's who, and we get everybody well habituated. We give them about 30 days just to live and hang out and get used to their new environments. And then what we are doing to study the response to Ebola is we're using a proxy, because I don't have a BSL-4 facility and I don't have BSL level 4 training. Now, some of the experimental infection work in Ebola viruses are done in those facilities. They're done in Germany, they're done in the US. That's beautiful work. It's not what I do. I'm working with wild-caught animals that are coming in with whatever they've got, and we are trying to understand in as close to the real world as possible what these bats want to tell us about how they respond. So we use something called an Ebola virus-like particle, or an EVLP. And it kind of sort of looks like Ebola. The filoviruses are like little pieces of string, and it's got all these little glycoproteins stuck on the outside. It's like the spike protein in coronavirus. So I think we've all become armchair epidemiologists because of COVID. We could all probably draw a coronavirus molecule with all these little spike proteins on the outside. In the filoviruses, we call that GP for glycoprotein. That's what binds to cells and allows that virus to get into the cell. So we have these it's like fake Ebola, basically. It was developed originally as a vaccine construct. And so it actually has 3 of the different proteins from Ebola that are studied on the outside of it. And so our bats, we give the bats a little injection. We give them a little vaccine, basically. And then, but they should see that and react as if, they've seen Ebola. And so we want to know, what does that mean to you? Tell us what so we're looking at immune proteins, for example, and we're looking at gene profiles. We're looking at what genes are being turned on or not turned on in response to this particular challenge. So, and then at 21 days, we give them another booster. And that's actually when we really see a much bigger response. And that's typical, right? So with our COVID vaccines, for example, we know that second shot gave us a bigger response. That's typical. So we give them this. We're also giving them different adjuvants. So an adjuvant is something that you can add to an immunization to try to augment the response that a body is having. So we actually have adjuvants that should tell the bat, wow, this is a viral infection. And then we have adjuvants that might say instead, this might be bacterial. And then we have a different adjuvant that's actually acting on a pathway called STING that all bats appear to have a genetic mutation in. So we're able to ask the bat a number of questions, and we're trying to really dissect the immune response that they are having to that particular pathogen. In our case, at the end, so we keep bats in total for 35 days after their first inoculation. We are euthanizing animals at the end of the study because we need to harvest the immune organs. And that's where, again, my sort of connection with Smithsonian comes into play, because if I'm going to euthanize an animal for research, that animal's body is going to go into a museum collection. We are collecting, we do what's called holistic sampling, right? Where we're taking a sample of everything. We have this, this picture of all the tubes that we collect for every bat, and we're doing swabs, oral swabs and fecal swabs, and we're preserving every tissue in every possible way, and we are sharing all of those tissues widely. So some of them go to my collaborators at CDC. Sometimes people write and say, "Do you have those samples? And can we have a new collaboration? Because I know you've got those in your -80 freezer in storage." So we do this holistic sampling. So we euthanize them because we need to get at the immune tissue to be able to explore sort of the cellular response to these infections. I also say that the species that we are working with are exceptionally common and not threatened in any way. They're these like peridomestic or synanthropic. So we're very conscientious about which species we use in terms of the ethics of what it is that we're doing, and we've had clearances at all levels, from the NIH to my university to Mooney University to Makerere University to the Uganda government. We think really hard about the experience that the animals are having, and then about the value of the data that they have just gifted us with.
Yeah, and then in what types of applications would those specimens be used? In other research.
Yeah, so that's the really fun and interesting part to me too, is that we have those will eventually come back. Some of them will stay in Uganda. We're hoping to gift some of those to the museum in the capital, and some of them we might use for teaching in those contexts. But most of them will come back. They might be in my lab for a little bit, and they'll eventually go to Smithsonian. And we can use those right now. Actually, we're using my previous specimens from Uganda and South Sudan in some studies of the taxonomic relationships in African fruit bats, because we actually don't know them very well. And in fact, what we consider one species it's our main study species. It's called a little epauleted fruit bat. And you have it in hand, and we look at the reference keys and we say, yep, this is what this bat is. But then when I have that bat in hand, sometimes I'm looking at this collection of bats. And then when I have them back at Smithsonian, and I might have their skulls out or be looking at a gene sequence, I think there's more than one species in there. And it's cryptic. And so we're actually starting to explore that. And here you have this lineage of bats, these epauleted fruit bats, and some of the relatives that keep getting pinged in terms of relationships with Ebola viruses. So in this lineage of bats, right, there's been PCR evidence, so genetic evidence for Ebola virus in them, and some other bat lineages as well. But here's this bat lineage that may play a really important role in multiple species in Ebola. So Ebola might not have a single reservoir host. It probably has multiple reservoir hosts. And some of those reservoir hosts might be in this lineage of bats that we actually don't even understand the species delimitations for. So these specimens are being used. Actually, we're starting to work this up now to help us try to understand the taxonomic relationships between all of these different animals. And, taxonomy really, really matters. So I was on a review paper that we published last year with a whole bunch of colleagues. It was led by Dr. Natalie Weber, a German scientist. And we reviewed all of the African bat work with all the African bat virus work that had been done through 2020. And so there's over 80,000 animals that have been sampled. And in many instances, the names were not given, or they weren't right, because it's like this One Health concept, right? It's like the people doing the work weren't collaborating with ecologists or with in-country scientists. And if you find some interesting new pathogen that might be significant for human health, you really need to know what bat that was from. And so in my mind, that also means we need to be, when we're doing some of this work, we do need to be vouchering. We need to be collaborating with museum communities, because those are the people who do that for a living, right? And then on the health metrics, so in the field, we sometimes do health metrics. I have these little veterinary diagnostic tools. You take a little blood and you put it in a cartridge and you put it in the machine, and it gives you the bat's electrolyte values. Well, I'm not a veterinarian. And so I kind of sort of don't know what half of those numbers mean. So I collaborate with veterinarians to say, can you tell me what this means? Are they in like metabolic acidosis, and does that mean that they're in poor health? And so we started by talking about the specimens, right, and what value they have. I mean, that's just one thing. We are now being able to go into specimen collections, including older specimen collections, even things that have been sitting in formalin for years, and being able to pull out genetic sequences and even partial sequences for RNA viruses, right? And being able to ask questions, historical questions about pathogens through time and place, right? So museums near and dear to my heart, and they're just a treasure trove, like historical snapshots of diversity or what we understood about diversity at different times.
Yeah, that's really cool. I'm going to ask a naive question just because it occurred to me, and I'm curious. But how do you avoid getting sick when you're handling, wild caught bats?
These are really, really important questions, and so we all have PPE protocols, so personal protective equipment protocols, and we do things to keep ourselves healthy, and we do things to keep the bats healthy. And so, for example, the current global protocol now, put out by the IUCN Bat Specialist Group, is that when you are working with bats, right, you're always gloved. You're like leather gloved, and then nitrile gloves or latex gloves over those gloves. You wear a face mask, so bats can get COVID. And so some species of bats may be able to have been experimentally shown to be able to get COVID. What we don't want to do is give it to bats in the wild and allow it to, evolve in them and then spill back again to something interesting. So we're trying to protect bats from anything we might have. And in general, when working with wildlife, that's a really important consideration. If you're working, for example, with great apes or primates, boy, we are so closely related to them, they can get our pathogens in huge ways. So some of that personal protective equipment is about protecting the wildlife species as well. But we have protocols in place, right, to try to mitigate the risk, right? So I think the risk is relatively low. It's kind of a needle in a haystack that you might encounter a pathogen, but the risk is real. And so when we are out in a village, we're not in our spacesuits. We're not in our personal air respiratory systems. We're not doing that because I just find that socially unacceptable. But we are wearing masks and we're wearing gloves, and we liberally apply bleach everywhere we can. And so we talk to people about that risk. Once our bats are in our facility, and we might be then collecting blood samples from them or handling them more. And then at the end, when we're doing dissections, then we get very, very serious about our personal protective equipment. So we're in Tyvek suits. We are in, ventilated hoods. And most of the tissue samples, unless there's some reason not to, most of our tissue samples get immediately preserved in something that inactivates pathogens. So there's a variety of different products out there. And they are just, they're amazing gifts really to the research community that you have these products that will preserve DNA and RNA, just sort of stop it in time, but also inactivate pathogens that might be in those samples. So we collect, most of our tissues are harvested in that. Sometimes we need samples to not be inactivated. And so we handle those, even more carefully. And, we have a whole protocols, we bleach down the tables at the end. We biohazard the little bags that the bats were in. We bleach our mist nets. It's not perfect, but we do our very, very best to try to mitigate those risks because they're real. Now, everyone in my lab has to have rabies shots. And that's great when we work in the Americas, right? Because rabies is a virus of American bats. It's actually not a virus found in bats in any other part of the world. I'm much more likely in Africa to get rabies from a feral dog. But, nonetheless, we all still have our rabies vaccines and any other travel vaccines that we can get.
We've covered a lot of really, I think, important and interesting ground. I've learned a ton today. I suppose, kind of a question as we move toward closing would be something along the lines of, what type of research effort would you like to see in the future? What areas of inquiry seem kind of the most interesting to you? This may be something that's, not necessarily on the near horizon, but kind of looking out into the further distance. What types of efforts would you like to see carried out in the future?
Sure. That's a great question. I think what we need to be doing moving forward is we need to move beyond in thinking about disease ecology and viruses that bats may host. We need to move beyond this sort of virus hunting, which was important for us to understand sort of viral diversity, but it is much more common now to try to be doing full sequencing of viral genomes, which is much more useful than having a partial gene from some virus and a bat that's not even identified correctly, right? So we need to be moving forward in properly identifying viruses and the hosts, including bat hosts that they might carry. So in the disease ecology work, that's what needs to happen. We also need to be doing longitudinal sampling. So lots of times we are out there and we catch animals and we collect samples at one time point, and then we go someplace else and we don't sit in a particular place and study what might be happening in those wildlife populations over time. That's really important information. And so I think I'm starting to see that happening more and more. And I think it's really, really critical work. I also think that as we get better at understanding spillover risks, and that we can come up with solutions that need to be pushed out, sort of ecological solutions, right, to preventing pandemics, to preventing spillover. We need to push out those possible interventions in specific ways to each different place, right? But we need to be working towards building the One Health community in each country with scientists from those countries, right? And we need to give them the tools that they need, and we need to come up with solutions that work to protect the ecosystem, to protect the wildlife, and to protect human health.
Great. I think that's a perfect note to leave it on. So thank you very much for joining me today.
Sure. It was my pleasure. Thank you.
And that concludes this episode of BioScience Talks. Just a reminder, the journal BioScience is published by Oxford University Press on behalf of the American Institute of Biological Sciences and is made possible by the support of our members and donors. Thank you, and talk to you next time.